My name is Markku Kelnanen, and I'm a plant biologist, but currently I work as a professor in biological spectral imaging here at the University of Eastern Finland. Hyperspectral imaging is a non-invasive technique, which means that we can follow anything that happens with the plants so that we don't harm them or disturb them. If we think of plants that are living organisms, so it means that they live, they have physiology, so all the cellular processes that makes life. So when anything happens so that it has then some consequences for the plants, we have to be able to monitor what is happening with the plant. And some of these symptoms are then visible. We can monitor them just by looking at them, and we can see that now there's something wrong with the plant. But with hyperspectral imaging, we can get then quantitative information about the symptoms. And this is why we are using it as a research instrument to have then more detailed information of what is going on there inside the plant. Plants are normally green because they have a lot of chlorophyll, but when they're stressed, they might be then accumulating other kinds of pigments. In this case, anthocyanin, which we see as a red color here in the youngest leaves of silver birch plant. We have here another birch plantlet, and if we look at the youngest leaves, we can still see the red color, but it's much more faint, and it's also present in some other leaves, but it's very difficult to estimate just by looking at it how much. How to find this out then is that we can image them with a hyperspectral camera. So we clearly have two classes showing that some of the leaves have a spectrum that resembles the youngest leaves that appear to human eye being red, and the other leaves are more or less green. Here we have the results for the second plant that was not so red, and we can see the same classes, but now we have a different pattern. We can clearly see that the anthocyanin is accumulating in many other places than on those leaves that we see it with our human eyes. If I think that how the IQ will change ways, the way we are doing research, or the way people are using spectral imaging currently or in the future, I think the main point is simply this: I can carry it with me. So I can take it with me out of the lab to the fields and ask research questions over there, or simply use it as a monitoring tool for different kinds of environmental problems or crop stresses that are there in the real world.